After the primary hemostasis comes the secondary hemostasis, which is also called coagulation. What is coagulation? When the soluble fibrinogen converts to fibrin, and these fibrin filaments, this fibrin will make the clot strong, and this process is called coagulation. Basically, this process is because the fibrin is a part of plasma. So basically, this process, the coagulation process, is the part of plasma, not the part of platelet or any other cell. There are two pathways for coagulation. First, intrinsic and extrinsic. So, what is the difference? Because initially they did not know how this coagulation happens, so they named these extrinsic and intrinsic pathways based on their findings. First, they found that some extrinsic factors or tissue factor or some extrinsic factors, when they add these tissue juice or tissue factors to the blood, they will make the blood clot. That is why they named this extrinsic factor. And in some cases, when they take out the blood and they put in a glass tube, they found that the blood is clotting. So they thought that maybe there are some intrinsic factors which are helping the blood clot. That is why they named it intrinsic pathway. So that is how the extrinsic and intrinsic pathway begin. So the normal blood clot process or normal hemostasis is called extrinsic pathway, and intrinsic pathway happens in the tube, in glass tube. When we put blood in glass tube, they form the clotting. This pathway is called intrinsic. Intrinsic pathway is not present normally. This intrinsic pathway starts with factor 12. Inactive factor 12, when comes to here and it gets activated, factor 12. This is now active. We'll make active as red. And factor 12 activates factor 11. Factor 11 is now active. Factor 11 activates factor 9. And this factor 9, along with, because this factor 9 is present along with phospholipids. Where are the phospholipids present? Phospholipids are present on this platelets membrane. So factor 9 in presence of factor 8, which is factor, remember this factor 9 is called anti-hemophilic factor B, and factor 8 is called anti-hemophilic factor A, because these two factors are important for the disease. So that is why we need to remember this, and we will make video in next phase. In presence of factor 8, calcium, remember the platelets also have these dense granules, also secrete calcium. These calciums are important here. So this calcium and factor 8 and lipoprotein. In presence of these three, factor 9 activates factor 10. this is active. And factor 10 activates prothrombin to thrombin. This is also active. And here begins the real work. So these thrombins are very active, activated thrombins. They now activate fibrinogen to fibrin, and this will be the fibrin monomers, fibrin filaments. These fibrin filaments will deposit, will make some black color fibrin filaments. So these fibrin filaments will deposit all over these clots. And what else this thrombin does? This thrombin, along with activating the fibrin, they also activate fibrin stabilizing factor. So these fibrin stabilizing factors will cross-link these fibrin monomers and they will make the mesh. Now this is become the secondary clot or secondary hemostatic plug. So this is now become secondary hemostatic plug. Now what is extrinsic pathway? So extrinsic pathway involves a tissue factor, TF, tissue factor. These tissue factors are secreted from, or these tissue factors are present on outer membrane of this endothelial cell or the subendothelial cell. These are also present in epithelial cells. So when after the rupture, factor 7 binds to tissue factor. This factor 7 gets activated. So now this activated factor 7 will activate factor 9 and also factor 10. Prothrombin to thrombin and fibrinogen to fibrin, these are the common pathways, and this is extrinsic pathway. This comes the extrinsic pathway, and these are the intrinsic pathway. So extrinsic pathway is very fast. That is why this is the important pathway which is present in normal condition. Now the blood clot is formed and the clot is very strong and the blood flow to the outer is stopped. So blood flow is stopped, and this is called hemostasis. Hemostasis, the blood clot is formed. Now what happens after the blood clot is formed? Actually, another thing I forgot to tell is this factor 5 is present here. Prothrombin is converted to thrombin by factor 10 in presence of factor 5. So the factor 5 is present here. So now we have known that all these factors which are secreted from alpha granule and dense granule or delta granule are used in different phases of cascade. So these factors we know are already used. What is left? The left is platelet derived growth factor. What is the function of platelet derived growth factor? These platelet derived growth factors which are released from activated platelet help in growth of both endothelial cells, also extracellular matrix by the growth of epithelial cell, the growth of sub-endothelial cell. So these platelet-derived growth factors help in growth of endothelial cell and some sub-endothelial cells surrounding the rupture, surrounding the damaged cells. So when these growth factors act, these cells will grow back. These cells will grow back. When these cells are growing to be finished, when these cells are finished growing, these clots need to be dissolved. How these clots gets dissolved? So in the beginning I told that the normal endothelial cell, they secrete nitric oxide, prostacyclin and ADP inhibitors. So now they are healthy cells. They will inhibit these factors, so they will act normally, like I said in the beginning. So the platelets surrounding this will be now inhibited and other factors will be now inhibited. And these cells, there are tissue plasmin activator. This will act now. This will be secreted by this endothelial cell, and this activator will activate plasmin, and plasmin will degrade this fibrin. And after the degradation of this fibrin, this will be dissolved, and this is called fibrinolysis. After the dissolution of fibrin, this will become normal. These cells, endothelial cells, have grown, and this is now normal. So plasmin also inhibited fibrin, degraded fibrin. Now platelets are going back to inhibited condition. And now platelets, if you see these round or discoid platelets, that means they are inhibited platelets, and other factors are inhibited by endothelial cell as previous. So this is the normal hemostasis process. One thing I need to tell, that this PF4 regarding the vaccines against this COVID-19 or coronavirus, there are some reports which shows that these AstraZeneca AZ vaccines are forming blood clots. So we have seen that they are activating clots through PF4. They are helping secretion of PF4 from platelets, and this PF4, they bind to heparin. PF4 will bind here, and they will inhibit the binding of antithrombin. Antithrombin cannot deactivate thrombin. So now the thrombin is active, and the thrombin, now the thrombin is active, and the thrombin will activate fibrin, and they will make clot. So that is how the basic mechanism, how the AstraZeneca vaccine is activating clots. In our next video, we'll talk about different diseases related to our hemostasis, and that is all about hemostasis. Thank you for watching my video. Bye bye. See you in next video.